So, good afternoon. I'm Fausto Rodriguez Zapata. I was a former research assistant here at CIAT. After working here in bioinformatics, I went to the Universidad de los Andes, and I made my master's degree there on the coca plant, on the genomics of coca plant. And then I got my PhD in NC State about genomic insights on the role of phospholipid metabolism and soil phosphorus availability in mass adaptation to highlands. So I will be presenting my work, two chapters of my thesis that I made on my defense on May. So, no, that wasn't. So modern maize has been the result of domestication, from early domestication of lowland, the lowland wild relative, Sea maiz subspecies Parviglumis, and then through its range of expansion, through the history of expansion, got into the highlands of Mexico around 6,000 years ago. Early domestication was about 9,000 years ago. So here it coincided with another kind of wild maize relative that has been adapted to the highlands, and that is called Teosinte. This is Sea maiz subspecies mexicana. So it has been shown that introgression from the highland adapted material is present in today inbreds that are cultivated in the United States. And for example, there is specifically one of these introgression helps in getting early flowering on temperate places compared with the lowland alleles. So two wild relatives made the current cultivated maize, and then this adaptation to temperate soils allowed maize to go overseas and now be the most cultivated plant in the world, and mostly on temperate lands. Most of the current hypothesis is that most of this adaptation to temperate latitudes might have to do with introgression from Mexicana. So the work in the laboratory is specific on biochemistry and on plant nutrition, and so we work on phosphorus, nitrogen, and also we do some metabolic mix work on specifically on lipids. So we had this hypothesis that maybe the metabolism of phospholipid might have helped maize to adapt to higher altitudes because the form and function of the membrane depends on the lipid constitution. Different lipids have different kind of forms, and this allows to modify the fluidity of the membrane. So in response to collant, that has been already shown. For example, in the roots of maize, one specific kind of lipid that is the phosphatidylcholine that is used in the bilipid layers gets reduced in order to increase the fluidity of the membrane and an adaptation to cold. So we thought that a similar thing might be happening in the highland maize. So on the other hand, these phospholipids are rich in phosphorus, and the soils at highland altitudes in Mexico are frequently low in phosphorus availability, especially andosols around the Mexico Valley have a high phosphorus retention. So another part of the previous hypothesis is that phosphate remobilization might play a role in adaptation to those Mexican highland soils, liberating releasing the phosphorus from the lipid membrane for use in the general metabolism. So we wanted to use this genomic information to address these two questions: is plant phospholipid metabolism contributing to maize highland adaptation, and maybe there's different kind of soils and the availability of phosphorus in those different soils contribute to adaptation in the highlands. So the first part that I will be talking is about this paper that I am co-first author in 2022 that says an adapted Teosinte mexicana introgression modulates phosphatidylcholine levels and is associated with maize flowering time. So I told you that we have this hypothesis that the lipids, the modification on lipids will adapt the plants to cold. So the first thing that we did, and this was even before I came to the lab, these data were collected before on a diversity panel that covered all of Mexico and South America. Here I'm only showing you specifically what is happening in the highlands and lowlands of Mexico. And what we see here is that in highlands, this phosphatidylcholine is increased, which is counterintuitive, but is the contrary, which is the opposite case that was happening in the roots in cold. So we didn't expect this at this time, like 5 years ago. But we continue to see if not just if this characteristic was not only just a divergence that might be due just due to neutral evolution, and we saw if there was a signature of selection on this. That means that the divergence in phenotype between highlands and lowlands is beyond this limit of the neutral divergence between these populations. And you see that the divergence in phosphatidylcholine content and some lysophosphatidylcholines, those lipids, is well beyond what is expected by natural evolution. So we found evidence of selection at the level of the lipids. We also went to see if there was evidence of selection at the level of the genes. So we grouped the genes in phospholipid-associated pathways, and we look in a general level. We can see specific pathways associated with phospholipids that show evidence of selection by measuring the divergence in between the genes in highland and lowland populations. An excess of mutations in the highlands population evidence selection over highlands, and excess of genes over the lowland population is evidence that the evolution has gone beyond the neutral expectations for lowlands. So we see here in the first row that phospholipid modification has been one of the pathways that have been selected both in Mexico and in South America. So we also see that the level of the metabolic pathway that there is selection. And now we wanted to see what specific candidates might be associated with that adaptation. So this was in collaboration with UC Davis, and Dan made this scan that consists of, here is the genetic PCA, so we see the diversity condensed into principal components, and the first principal component is highly correlated with elevation. So the variation in this elevation then is associated in a genome-wide association study with all the observed variation in this population that is a different population, a different diversity panel that has been collected throughout Mexico. So this is about 3,000 lines, and we found that the genes responsible for the synthesis, in this case, CML LPCAT of phosphatidylcholine, and also the degradation of phosphatidylcholine, in this case, HPC1 here on the chromosome 3, show evidence of association with elevation, and this is beyond also a neutral expectation here. So we have selection at the level of the metabolites, the gene pathways, and we have now candidates specific candidates that might be responsible for that selection. So this is just a genomic association. So we have to prove that the mutations on the variation on those genes produced a change in phenotype. So, and an additional population that was by parental cross between a highland and an inbred lowland maize. We made a QTL mapping, and we got a hit that the marker was in the middle of the coding sequence of HPC1, that is a phospholipase that catalyzes the degradation of phosphatidylcholine. So this variation is associated with the accumulation of phosphatidylcholine in the highland material, and the lines that get the highland allele had higher content of phosphatidylcholine. So it reproduced what we observed in nature, this cross. But additionally, in association with Cold Spring Harbor, and that was mainly the work of the biochemist at the laboratory, Alison Barnes, we produced CRISPR mutants, 2 alleles, one allele that was a missense and the other allele that was a shift. And the 2 alleles phenocopied what we observed on the natural variation in our population. So we proved that variation on this gene, although not exactly the same allele, reproduced the accumulation of phosphatidylcholine like in the highland material. So we have here the accumulation in the highland material that is contrary to what we have been expected from the work on roots. So we wanted to see what may have different phenotypes that it was showing on the plants. So what we do was scanning not just for the we're scanning not just for the additive effect of variation in HPC1, but we scanned for the GY interaction throughout the genome. And we saw that HPC1 is one of the big peaks for GY interaction, which means that the effects are swapped between highland and lowlands. So in lowlands, HPC1, the variation of HPC1 is associated with later flowering, while in highlands, it accelerates the flowering time, the same allele. The highland allele shows delay flowering in lowlands and early flowering in lowlands, compared with the inbred here. So that was one thing that is specific to the highlands and shows the typical pattern of local adaptation. In addition to that, in the context of the genome, we see that there are additional populations where it has been shown that the variation on HPC1 is associated with an early flowering of about 1 day. That is a lot, because the greater effect that has been shown is here in chromosome 4, about 3 days. So it was 1/3 of what we have found in chromosome 4. And the variation that we observe here was limited to HPC1. So you see a linkage disequilibrium block that is specific to the coding sequence, while the promoted region is out of linkage disequilibrium, so it evolves independently. So in addition to that phenotype, we confirmed that the alleles that we see on the highlands of Mexico, Mexican highlands here, Guatemalan highlands, are shared with the Mexicana, so the source, the potential source. And we proved by statistics of ABBA-based statistics that the direction was from Mexicana toward cultivated maize, while it is mostly absent in the lowland wild and the lowland wild relatives. And in addition to that, we went over data of flowering in the greenhouse, and so association between the expression of HPC1 and the time of flowering in long days. So it is the flowering in temperate regions. So Higher expression of HPC1, also what means more degradation in theory of the phosphatidylcholine, less phosphatidylcholine, increases the flowering time in the temperate regions. So that works not only in temperate but also in the tropical material. And again, the color here represents which kind of population we have here. So this is the population that the allele mexicana has been introgressed to, that is mainly temperate material. And here is the tropical material where the lowland allele is most likely present. So we have evidence at the biochemical level, we have evidence at the genetic level. So we wanted to see if this association of flowering time had like a possible molecular mechanism that has been mediated by phosphatidylcholine. So we know that zeta-CN8 is the florigene in maize. So this florigene is regulated by phosphatidylcholine. As a matter of fact, when it was the first time that this gene was cloned in Arabidopsis, it was called ethanolamine binding protein, because there is a pocket here that allows for binding with lipids, and in our case, it binds better. This is just a model. It binds better with phosphatidylcholine than other lipids here. So the biochemist at our lab made a construct, expressed it on many things, but what ended up working was in yeast, where here in yeast we show the positive control. No, the positive control is below. So the positive control here is when you incubate the construct of CCN8 with this standard phosphatidylcholine, and then you pull out the protein out from the solution, and you send this to mass spectrometry to see specifically what lipids were pulled out from the construct that you expressed there. So with the positive control, you have here the phosphatidylcholine that we added, and here we have the construct that come from a highland allele. So the highland allele binds to CCN8. So we have a plausible mechanism that differentiate the kind of association, the kind of functions that the phospholipids does in mice. And roots, it seems that associated with cold and increased membrane fluidity comes from lowering the amount of phosphatidylcholine that make the the bilipid layer. While in the leaves, there is a different history, where the adaptation to highlands is implying that there is more of these phosphatidylcholines that might be accumulating and binding to the flower, the florigene, the florigene gene, and is inducing the flowering time. So this has been shown in Arabidopsis, where phosphatidylcholine binds. This is a different lipid, but in Arabidopsis, this lipid binds to the florigene and activates the FT gene for producing early flowering. So that was a nice story. And here is the summary of the investigation. This is that we wanted to express through this drawing that one member of our lab did, Andy Kerr. So in the lowlands, we have alleles that come from Teosinte pargibrumis. We have more lysophosphatidylcholines than phosphatidylcholines, and the phospholipase that we have is more active, which produces this step of the reaction, the degradation of phosphatidylcholines. The lack of this phosphatidylcholine might be causing late flowering of the lowland material when it's grown in the highlands. What is happening here on the highlands of Mexico, you see everything is flowered, is that we have the introgression from wild material that was probably adapted to highlands. We have accumulation of phosphatidylcholine. The phospholipase HPC1 is less active, and we have a mechanism that the accumulation of phosphatidylcholine causes this early flowering. And this allele is widespread in northern material. So that was a nice story, and it required like 30 co-authors to work this for more than 6 years. So I was happy to participate on that. So the second part of what I'm talking today is the effects of highland maize chromosomal inversion in flowering time, gene expression, and phospholipids. So I want to come back. Oh, well, we have again this. Sorry. We have again this typical climb of allele frequencies through elevation, where we have this inversion in the chromosome 4 that is mostly fixed at elevation greater than 2,500 meters in Mexico. So in the highlands of Mexico, this inversion is fixed. When I was talking about the scanning for G-by interaction, the other region in the genome that was absolutely fixed, that was a big peak for gene by environment interaction respect to with elevation was the inversion for M, around between 180 megabases in the chromosome 4. So these sites here are associated with flowering time and getting more earlier flowering in the highlands and delayed flowering in the lowlands. But also it has a similar pattern of gene by environment interaction, where it's contributing to higher genes in the highlands, which HPC1 didn't show. So again, as might be supposed, the inversion for M is present in the Mexicana. Here is this is a graph of synteny. So the gray parts show that there is collinearity between the genomes of Mexicana and, in this case, one highland cultivated mice, PT. That is the one that I used previously in the biparental populations. So there is collinearity here. So this region of the genome is inverted relative to B73 that was the recurrent parent in this kind of cross. So in order to test for the hypothesis that the inversion specifically is associated with local adaptation to highlands, and if that adaptation has something to do with phosphorus, what we did was isolate the inversion on B73 backgrounds using isogenic lines, where at the end we selected for heterozygous and we produced by self lines that were segregating for the inversion here. So at the end, the control of my experiments were siblings, where I have a sibling that was homozygous for the reference karyotype, and the other was homozygous for the inversion. This was done in Pennsylvania, so it was a temperate conditions, similar to, in this case, what the conditions in the highlands. That was the best thing that we could do. We measured phenotypes, and the thing that we showed about the inversion for M is that it produced higher plants and earlier flowering, independent of the conditions of phosphorus. Either in plus phosphorus or minus phosphorus, the effect of the inversion was faster development. We also saw that this was the case just for the flowering time, for the flowering time phenotypes, not so much for the other phenotypes that we measure here. But the effect of phosphorus was in every case either reduce flowering or increase yield or biomass. So that was what we expected. But it shows that there is not a specific gene by interaction, so not specific that this is not contributed in this manner through phosphorus to the adaptation to highlands. In the next part of the experiments, we sampled leaves, because we already know about the contribution of leaves of the gene expression in leaves to flowering time. We sampled leaves on the two conditions, plus phosphorus, minus phosphorus, in the two genotypes. So using this data, we were able to confirm that the inversion that we have selected through 8 generations was really selected, and what we use a marker, the Cas-Berry marker from the SNP, that we show confirm that we have our inversion and a little bit more of chain integration around the part that we have. So it's like twice the size of the inversion. This is like 15 megabases, and we have additional 30 megabases of the highland genome there in our lines in an experiment. Regarding the expression, the greatest amount of variation here is explained by, I don't know if you can see, but the different treatments, the treatments in high phosphorus tend to be to the left of the plot, and low phosphorus tend to be to the right. So the greater amount of variation is due to the fertilization status of the soil. And the second axis of variation is associated with the age of the leaf, where you see the top is the younger leaves, and the bottom is associated with the older leaves, the leaves that are more senescent. That's why I put it like yellow here, that have more senescence. So we analyzed the gene expression through a mixed model for the effect of the leaf, the phosphorus status of the soil, the inversion, and the interaction, the gene-by interaction. What we found is in the leaf that this is a nice result. One of the genes that were most regulated by the leaf age is that in older leaves, the genes associated with flowering time has been suppressed. So CMMF4 is a matchbox gene that is the gene that is expressed in the leaves that is more correlated to flowering time. So we were on the right track following the genes that we were interested in the phenotypes that we have measured. So this gene expression is the gene expression of this matchbox gene. Here is one of the first hits. And you see that the gene expression around all the genome is significantly different between the two adjacent leaves, old and younger leaves. These also show up in a gene ontology analysis of the biological process that were more likely regulated as a whole by many genes. And we see here, this is downregulation of chlorophyll because the leaf is becoming more senescent. And also here is downregulation of carotenoid by synthetic pathways. So that's one of the results for leaves. We also have the second axis of variation that is the phosphorus response. We show that is also widespread. It's about, that was 14,000 genes out of 24,000 that were expressed. In this case, for the phosphorus response is 10,000, half as much, half the total amount of leaves that is expressed in the leaves. And here the response was the upregulation of genes associated with phosphate response. For example, purple acid phosphatase, and this is the PI deficiency induced long non-coding RNA1 that is the master switch of regulation of phosphorus response. So this showed also that the design was good enough to detect the two biggest causes of variation. Again, phosphorus response is the first time that appears in the gene ontology annotation. And as I told you before, the response is throughout the genome. In contrast to that, the effect of the inversion is restricted to the introgression that we have brought into the B73 background. And the highest and the most regulated gene in this case is a gene that was downregulated in the inversion. Is a Genomic transcription factor. But really, it's not a transcription factor. Genomic has a part that binds to the DNA, and that's why the annotation of transcription factor appears. So that part is, it binds the DNA, but it has a domain that has histone demethylase activity. So it's modifying the demethylase, and it's modifying the accessibility of the DNA for the RNA polymerase. So it's modifying the expression around the genome in a sequence-specific way. So in addition to we have three different genes overlapping here on the same loci in BN73. This one that says Jumanji2, this corresponds to these three genes annotated here. In addition to be a tandem repeat, there is an additional, there's an extra member in the B73 in the cultivated mice, on the lowland cultivated mouse. So this difference in gene expression might also have something to do with this lineage expansion that went into the cultivated lowland mice. So that is another clue of a candidate that might be something to that might be associated with the difference in phenotype occasioned by the inversion 4M. And the nice thing is that the Jumanji family of transcription factors that also have the demethylases, these demethylases are associated with repressing the flowerage, the FT, the FT that was the gene that I was talking about in the previous chapter. So these Jumanji are repressors of flowering time. So it's nice that when the repressors are repressed, the flowering time is early, so it accelerates. So that's the final result here. We have a specific expression caused by the inversion. It's also shared by the integration, but in this case, we need more data to disentangle these two results. We have the classical response to phosphorus, a typical response of older leaves, and suppression of flowering time genes throughout the age. So these were results that I've been working this week because I have to. I brought my thesis dissertation, but I want to publish this with one submittal and plus. In addition to the gene expression data, we also have mass spectrometry data on the lipids. And I did a similar statistical analysis that I did, and we also see the classical response to phosphorus starvation. That means if you are under phosphorus starvation, you don't want to use lipids that use phosphorus as a head group. Instead, you want to use lipids that use sugars as a head group. So these phosphoglycerolipids are decreased under phosphorus limitation. The glycerolipids that don't use phosphorus are increased. So these are decreased, these are increased. And there is also accumulation of triglycerolipids, the triglycerolipids that are oil is accumulated when there is phosphorus deficiency. So that is a response that we saw throughout all leaves, but in the other leaves specifically, we saw an effect of inversion 4M, where the phospholipid phosphatidylethanolamine has been decreased, and the neutral lipids, the oils, have been increased. So this is also the hypothesis that we have here is that the inversion 4M plants have a lipid profile that is more like a plant that has been already under phosphorus deficiency. So this looks a little bit like the plants that are under phosphorus deficiency, even if they aren't. So this might be a way to see this. And also, I told you that phosphatidylethanolamine is binding to the florigine gene, to the FT gene. So this also might be, the lack of this might also be regulating the florigine, the time of flowering. So there are two routes that inversion 4M might be causing adaptation to highlands. So in summary, what we have here is regarding phenotype, inversion 4M, this is the first time that there is actual like isolation of the inversion and showing that the just inversion or mostly just inversion causes a decrease in the flowering time in maize. So the inversion 4M makes plants grow faster, grow larger and faster. We can detect the typical, classical response to phosphorus starvation. And the gene expression, we also so the same thing reflected on gene expression, and we have a candidate, the Gen1G, that might be associated with the illness flowering times. And it has a corresponding change in the lipid profile. So that was the two things that I prepared for now, but if you want me, I can keep talking. I don't know how much I am about the time.
We still have time, but I'm not sure if people has questions. I have questions, so any question? So I have one.
Okay.
So you were showing this G by GWAS?
Yes.
So how many different environments were tested, and how you combine the data to do the G by GWAS?
Yes.
That one.
Yep. So it's supposed to be here. We have the 3,000 different land races of traditional varieties that went into this plot. But we have here, that was in black. In red, there are 32 stations, CIMMYT stations, field stations, where the population was screened, how do you say, phenotype, where the population was screened. So, and the population, you know, this is a special kind of population because the things in highlands don't want to grow the things that are there in lowlands, so you have to, what is there was an F1 from a locally adapted variety with a different donor. So that's the kind of population that we do this. We do this in 32 different, well, the people did this in 32 different locations. And the statistics way to do this is just looking for significance on the GY term, and the same thing like a GWAS. So you establish that in the mixed model, and the mixed model that describes the variation. Yeah.
So from that one, so from 32 locations, did you also include any data from weather, climate change?
That's another part. That's another part. So yes, this population, this is a nice population that you can make genomic environment associations. Mm-hmm. So there's already published a genomic environment association with these land races and the first hits are associated with precipitation. Precipitation is the main thing, and well, evidently, elevation is the first thing, and after the elevation, it's precipitation. Many of the parameters from World Bio came of precipitation, but also there is a data set for well relatives, just for teosynthesis, highland teosynthesis, lowland teosynthesis, and that is something that is not yet published, but it has been studied here. And another additional chapter in my thesis was exactly that. I can show it here. How can I do this? No, I don't have it here. I deleted it. So I have a map of phosphorus throughout Mexico, and I made the map of phosphorus. I made the predictions of the map of phosphorus throughout Mexico from point profiles, from 7,000 profiles that were in the Mexican database that is not included because I don't know, because it's in Spanish, I think. So I made that. I made the predictions on the phosphorus. I made the association with the observed variation, and I have another set of candidates that are auxin related transcription factors. Yeah, I did the same thing. So yes, the idea was to do that, but was the correlation between the experimental and predicted phosphorus is just 0.3, so it's not great. Yeah. That was another chapter of the thesis that I didn't include.
And, thank you, Fausto. I think it was a very interesting seminar, and I have from the breeder's perspective, a lot it has been maize is classified between maturity groups. So the way the maize breeders do breeding is all related to the maturity groups. is any of these genes related to these different maturity groups? Because they are changing quite the pattern in terms of flowering time. So you have early flowering adapted to
Yes,
highlands.
but I don't know if the maturity groups has something else to do with the photoperiod sensitivity. That is a different thing,
Yep.
but I'm not sure. But that has been shown that to be responsible of one single gene that makes teosinte sensitive to the photoperiod, so it doesn't flower in temperate. So that sensitivity is lost in the cultivated mice in temperate regions. But that's an independent thing from these kind of genes that I'm talking here.
Yeah, those.
I'm not sure if this is the same, but yes, those are independent. At least it seems to be the case.
Okay. So, any other question?
Okay, very impressive. There is now some kind of strategy to reduce the height of maize and then to boost yield. Like, what's your data kind of will predict if a strategy is to reduce height of maize?
Okay, because there are different ways in that you can make a small corn plant. So you have to make a small corn plant that preserves the yield that you have already. So that's the difficult part. So most of the genes that are well known as the brachytic, that is, I think, is homologous with the dwarf genes used in the other in wheat and rice. So it's I think it's an auxin related and something an auxin transport or something related with the auxin. But that is another thing that they're trying to do is looking for alleles that are different to that. I don't know if that has to do something to the lowland plants in the highlands are they are just smaller. So yeah, I show you one of these in those photos. They grow smaller. So that's kind of things that the kind of genes that we show here that are associated with height are sensitive to the environment. So I don't know if that would be the kind of thing that we observe here. If that would be useful. What would you like is something that the brachytic that is dominant and is not that has not an interaction with the environment. And there are other genes. As a matter of fact, in the lab we isolated one Mendelian that is very nice. In this population of PT by 73, we have a plant that is something that looks like brachytic mutants, but we are not sure. And I'm working on those data to see if it maps back as an allele of brachytic or it is something different. The developmental biologist back there in North Carolina, he told me that what he sees is different because the internodes, they preserve some kind of proportion, so that would be a nicer kind of allele because the development is more similar to the high yield populations. So that is the kind of allele that we will be interested in, and that might be one. But as a strategy, I wouldn't go with the elevation-dependent genes variation. Yeah.
Thank you for your presentation. I am very interested with the integrations. So I was wondering, you identified an integration share in a genetic group of the highland genotypes, right?
Yeah.
So I was wondering if occurred one time integration, is the possibility that in the genome there is another integration located in the genome. So how to verify that this integration is the integration that is functional and is not interacting with the other integrations that is in the genome?
So there are many ways to that, but one simple way is to see that our integration has the selection that is correlated with the inversion for M. So everything here is mostly has the same genotype, and there is some kind of, I know this library is the library with the smaller number of reads. So this is lower quality and it has more noise. But in general, what I predicted to have the integration has the inversion and has a little bit of integration here, and it is all correlated in a single linkage group, linkage disequilibrium group. But otherwise, the background is random. So the background here is like, yes, like a random variable at every place. There is no other loci that are correlated in the same way that in the disequilibrium with this. So we know for sure that this is the thing that is making at least this integration is the one that is making producing the effects that we observe in expression and the genotype, and the rest is randomly distributed. The other thing that we did that it was done previously in a work on this population, that it was not just a single parent. In this case, I have Michoacan 21, but there is an additional PT population on this. I did my experiment here because that was the one that I had seeds, and I can plant at the time. But we have a new population with a different parent that has the same inversion, that has gone through the same breeding scheme. So the two different alleles independently producing the same kind of effects are less likely. So that was the other way that we can work on that. And yes, and you can tease out and that with that population also this part, if it was actually the inversion or the shared integration here that is responsible for that too, because it's different. These different alleles on the other line.
I have another question. So you identified the integration according to the read counts?
No. So in the reading design, what we did is we made the cross. We selected here. Here we use a CAPS marker. A marker that is derived from a SNP that mark the inversion. We know the highland and the lowland lead. So I always selected the plants that were heterozygous for that marker. So I knew that it was it has the inversion 4 and it was heterozygous. So I chose the heterozygous, and just before this generation, I did a self of those heterozygous. So I have a ceiling that with that marker, I genotyped and I found that it was homozygous for the control karyotype and homozygous for the inversion. So I knew that. So after that, I sequenced the genes by RNA-seq. So these are the 20,000 markers corresponding to the 24,000 genes expressed. And each point is a region of markers derived from the RNA-seq data. So I know which was highland or lowland at each point, at these 20,000 points throughout the genome. So I know the genotype on every single place here at the resolution, at the genes. So it was at the genes that were expressed. So this is a nice way to do this. But yes, it's expensive because it was $20,000 for all the sequencing. Okay. For 64 libraries, yes.
But you use it a reference to...
Yes, B73. The reference is B73, and in my cross, the yellow thing is B73, so this is the same thing, that is the reference for maize.
Okay.
So that is also a nice thing about this.
Because I was thinking that if you, those samples that comes from other background, different to B73, maybe in this region have a longer segment or a different information than the B73.
Yes, that happens, and you can do the other way around, but I don't have the exact same part. Well, at this point, I didn't have the parental, but I do now have the parental. I can do the switch and see if something else appears that hasn't been mapped. Because we know, as I told you on the Genomic cluster, that it is copy number variation. So in this case, I have two copies here, but in PT, that I now have that data, there is only one copy and one copy Mexicana. So I can come back and do the mapping of the other way around, but yeah, that might be, but I don't think I will do it now.
Okay, thank you.
No questions?
From an ethnical point of view, is there any kind of separation between the different populations? from the kind of the different ethnic population between the lowland and the highland?
Jeff Rosibarra did a nice work the other day. Yes. So he made a nice work in the Yucatan Peninsula, where he compared the divergence between lowlands and highlands. Well, Yucatan is almost flat, but they're close to Chiapas. There are some mountains. So there are highland and lowland maize. And he compared the divergence between lowland and highlands and he compared the isolation by distance in the flatlands in Yucatan with the isolations of different land races from different like indigenous populations, indigenous groups. So he saw that there was some genes that were more differentiated between different indigenous populations than what you would expect by distance. So that things happens. And he compared also the difference in language. He compared the distance in language, the genetic distance, and there is some effect there. But it was specific for some genes. I think he has like 16 genes that are candidates to that. I didn't do that because that's not in my scope, but I know that that happens. Yeah.
Okay, so if there's not any other question, I think that we can finish now. We have 2 minutes for 4. So Fausto, thanks for coming back and showing us the development of your PhD thesis. And yeah, we look forward to see the next paper.
Thank you.
Thank you, everyone.